Yes, all right. Welcome back to Anonymous MD, the place where facts don't care about your feelings and where we find the bias in science. So take a moment, share, like, subscribe so that we can continue to use our evidence-based principles and counteract the recent epidemic of statistical shenanigans. And in this case, we're going to see an example of how governments are interpreting and using data to make policy and regulatory decisions. So usually when I scan for articles to share with you, I'm using Apple News or Google News because that generally reflects what the majority of people are seeing on a day-to-day basis, you would think. Ultimately, I want to use my skills to fish out the scientific sources cited in any given article and analyze them as objectively as I can. So today, let's look at this article from Reuters dated February 23rd, 2022. The headline reads, AstraZeneca signs deal with Canada for 100,000 doses of COVID drug. The article starts by stating that Canada bought 100,000 doses of a medication for prevention of COVID-19 in some high-risk patients. The article goes on to read that this medication, called Evusheld, is under review by Health Canada for use as a preventative treatment against COVID-19 for those who are immunocompromised. So here's a quote from a Canadian health minister, or the Canadian health minister, I'm not sure which one, who again references immunocompromised hosts as the beneficiaries of this treatment. Deliveries are said to arrive a month after authorization, implying that this deal between AstraZeneca and Canada is taking place during the pre-authorization stage of the medication. The article ends there, short and sweet, but let's dig in deeper and find the scientific basis behind making this decision to purchase 100,000 doses of a pre-authorized drug. An initial Google search highlights this FDA news release from December 2021. It quickly becomes apparent that the FDA issued an Emergency Use Authorization, or an EUA, for Evusheld for pre-exposure prophylaxis or prevention before getting the COVID-19 in certain adults. Criteria for use includes that the individual does not have COVID-19, has not recently been exposed, and is either immunocompromised or has had a very bad reaction to the COVID vaccine in the past. Here, the director of the FDA Center for Drug Evaluation and Research refers to this as an alternative prevention option, and the article continues on that the medication may be effective for pre-exposure prevention for up to 6 months. The FDA news release goes on to describe the mechanism of action of the medication in that it's monoclonal antibodies that are prefabricated and designed to attack the spike protein of the SARS-CoV-2 virus, thereby blocking the virus's ability to attach and enter human cells. The next paragraph provides us with the most useful information, and that's the source of data supporting this EUA. They use all the buzzwords when initially assessing the quality of this study when describing the PROVENT trial. They say it's randomized, double-blinded, placebo-controlled, which sounds very promising. What's more, they reported that Evusheld recipients saw 77% reduced risk of developing COVID-19 compared to those who received the placebo, which was a statistically significant difference. In episode 1, we discussed statistical significance, and if you need a refresher on the concept, I encourage you to go back and watch that video or reread about the concept. I couldn't find a PDF of the original study. I'm not sure why. Maybe you can tell me in the comments below or share the link. I did, however, find reliable sources that provide enough information about this study for the purpose of our analysis. Here we have an abstract from the journal Open Forum Infectious Diseases published in December 2021. This is an online and open access journal that is published by the Infectious Disease Society of America. New medications undergo 4 phases of clinical studies, the details of which can be reviewed on future videos. For our purpose today, we should understand that by definition, Phase 3 denotes the final phase of pre-market research, the results of which are submitted to regulatory agencies such as the Food and Drug Administration, or FDA, to evaluate a drug's effectiveness and its short-term safety. Phase 3 trials provide the basis on which medications are approved by the FDA for marketing and are therefore expected to be well-designed and randomized controlled trials. In other words, this is meant to be the phase that definitively proves that a medication is effective when compared to other therapies or placebo. Phase 4 starts after the medication has been approved and is on the market, which is the final step of research and is focused on identifying any long-term side effects or adverse events caused by the drug, thereby providing a final safety profile. The introduction of the abstract notes that the paper is reporting the primary Phase 3 study results of AZD7442 for pre-exposure prophylaxis of symptomatic COVID-19, the numbers and acronym representing the medication Evusheld. The methods section notes that PROVENT is a 2-to-1 randomized, double-blind, placebo-controlled study, which are the standard requirements for a quality randomized controlled trial, or RCT, and the basic elements required for an acceptable Phase 3 trial. Meeting this criteria is necessary for an RCT to be considered reliable, but in and of itself does not guarantee the reliability of a study. In other words, when studying the effects of a medication on outcomes, all good studies must be RCTs, but not all RCTs are good studies. The outcome measured was a first case of PCR-positive COVID-19 and symptoms, that is, confirmed symptomatic illness, up to 183 days or 6 months after administration of the drug. The results section shows a relatively large sample size of almost 5,200 participants. Large sample sizes are desirable and improve the ability of a study to detect true differences between the study groups. The validity of results increases as the study sample size increases, that is, the results become more accurate and less questionable. The results purport to show a 77% risk reduction in developing symptomatic COVID-19 with the monoclonal antibody versus placebo, and the p-value is less than 0.05 or 5%, making these results statistically significant. So, on the surface, we have a well-designed study with a large sample size, so we would be hard-pressed to argue the accuracy of these findings. It is notable that the abstract does not include the actual number of positive cases in each group. For example, how many of the 1,737 subjects given placebo developed symptomatic COVID-19, and the same for the 3,460 subjects who received the medication. Any academic and objective reader should notice this omission and wonder why these results were not highlighted. As we will learn, comparing the rates of outcome between groups is part of the fundamental analysis of research results, and excluding this information from the abstract automatically makes me question the conclusions being reported. In conclusion, the abstract notes that the primary endpoints were met and a one-time dose of AZD7442 demonstrated statistically significant protection against symptomatic COVID-19. Before we move on to further analyze this study, the funding statement should be noted. Sources of funding and conflicts of interest shed light on the funding bias of a study, whereby the results have a tendency to support the interests of any financial sponsors of the study. It is notable that this study was supported by federal funds from multiple U.S. government departments, including the Department of Health and Human Services and the Department of Defense. The following disclosures further reveal support and grants from several other global pharmaceutical giants, including Pfizer, Sanofi, GlaxoSmithKline, and Johnson & Johnson. Further down, we should notice that most of the researchers involved in this study are AstraZeneca employees and shareholders. These conflicts of interest are not surprising, since Phase 3 trials are usually sponsored by and studied internally by scientists from the manufacturing company. These conflicts of interest in and of themselves do not reflect the quality of research, but analysis should include an awareness of potential ulterior motives that, when present, provide a context for a bias analysis and reporting of results. In this case, the omission of critical data, the statement of funding, and the disclosures herein suggest to me that the abstract seemingly convincing data and the resulting conclusions are not sufficient, and that the authors may be cherry-picking information that sheds a positive light on their research. So, although this abstract seems to present a well-designed study with significant findings, further analysis is still required to evaluate the true reliability. In particular, and even though the results of the study are statistically significant, the critical information that is missing leaves me wondering about the clinical significance of the findings. To understand the concept of clinical significance, we first need to learn the difference between relative risk reduction and absolute risk reduction. Let's use the results of an example study to elaborate, in which 20% of the placebo group experienced a bad outcome versus only 12% in the treatment group. The relative risk reduction reports the reduced outcome in the treatment group as a fraction, or proportional to, or relative to the placebo group. In this case, the relative risk is 12 divided by 20, or 60%, which implies a 40% relative risk reduction between the two groups. Relative risk reduction is a technical value that we use to apply the findings of a study to other populations that have different rates and incidences of the same disease. Using it as a measure of the results and clinical significance are misleading, but this is usually the risk measure that is represented by researchers and thereby included in headlines and reported on by the media. The reason why is because they can show large decreases in the risks and imply a very strong effect for the intervention. In this AstraZeneca study, the 77% decrease in symptomatic COVID-19 cases is indeed a relative risk reduction, and the difference in findings are statistically significant and therefore reliable, and the medication actually made a real difference between groups. The problem is that this information does not shed any light on if this medication has any clinical significance and use in practice. To further understand this concept, let's use the example of a 10-point pain scale as an outcome of a study, which is often the endpoint when trying to determine if a medication reduces the extent of pain. If the medication reduces the pain from 10 out of 10 to, say, 5 out of 10, and the results are statistically significant, this would represent a 50% risk reduction in the outcome. 10 out of 10 pain represents the worst pain of someone's life and suggests that the patient is suffering. Since the burden of the untreated pain is so high, a 50% reduction to 5 out of 10 would provide noticeable relief for the patient and therefore has real-life applicability and would be deemed clinically significant. On the other hand, if the untreated pain is 2 out of 10 and the medication reduces the pain to 1 out of 10, the relative risk reduction is also 50%. But even if the difference is statistically significant, the patient is not nearly as likely to report pain relief from the treatment. That is, because the initial burden of pain was already negligible, this statistically significant 50% relative risk reduction does not have any practical effect for the patient and is clinically insignificant. So, both examples reliably report that the treatment leads to a 50% reduction in pain, but only one study has any real-world applicability. On the other hand, absolute risk reduction, or ARR, is the most useful way to present research results. The absolute risk reduction is the difference between the percentage of the outcome between the placebo group and the study group. That is, the percentage of events in the study group is subtracted from the rate of the same event in the placebo group. So, in our initial example, the absolute risk reduction would be 20 minus 12, which equals 8%. If we divide 1 by the absolute risk reduction, we can calculate a very useful value known as the number needed to treat, or NNT. The number needed to treat represents the number of interventions required to prevent one bad outcome. In our example, we would calculate 1 divided by 0.08 to equal 12.5, which means one bad outcome is prevented for every 12.5 people who received the treatment. To better understand this concept, let's go back to our pain scale study example. Reducing the pain from 100% to 50% or from 10 to 5 is a 50% absolute risk reduction. If we calculate the NNT, or number needed to treat, it would be 1 divided by 0.5 or 1 divided by 50%, the result being 2, meaning that the treatment helps 1 in every 2 cases that receive the medication. By contrast, if we reduce the pain from 20% to 10% or from a pain scale of 2 to 1, this is a 10% absolute risk reduction, and 1 divided by 0.1 or 1 divided by 10% would equal a number needed to treat of 10, whereby the treatment is only effective in 1 out of 10 interventions. Here we can see that the 2 treatments with the same statistical significant relative risk reductions have very different NNT values, which mathematically shows us the difference in their clinical significance. Any phase 3 trial results should have both statistical and clinical significance as the prerequisites for approval, That is, the treatment should show true effects that are not due to chance, and those true effects should provide clinical results that have practical implications. The abstract that we reviewed failed to report the absolute risk reduction and also omitted the rate of outcomes in each group that would be required to calculate the ARR. So, to complete the analysis of Provent, I found this source that provides the outcome rates between the two groups. We can see that the rate of first symptomatic infection in the placebo group was 1%, whereas it was 0.2% in the treatment group. Only the relative risk reduction is calculated here, but we can easily see that the absolute risk reduction is 0.8%. We can further calculate 1 divided by 0.008 or 1 divided by 0.8% to come to a number needed to treat of 125. In other words, we can prevent one new case of COVID-19 for every 125 doses of the drug that is administered. That means that less than 1% of recipients derive any benefit, and of the 100,000 doses purchased by Canada, they will prevent 800 cases of first-time COVID-19 infections. When we compare the more useful figures of the NNT to the 77% relative reduction rate that is reported, we get a completely different and much less convincing picture on the applicability of this medication in real life. In reality, the relative risk reduction is often used by study authors to report results despite its real-world limitations. In my opinion, this is the scientific equivalent of cooking the books and make results seem more favorable. I can use my training and experience to calculate and use more practical measures of efficacy, but a non-scientific observer would view a 77% reduction in the outcome as very effective. Before I share my final opinions on the purchase of this medication by Canada, there are two more very important flaws to this study that we must review. First, the news article and FDA website emphasize the use of this medication specifically for immunocompromised people. Yet, less than 4% of the subjects in the study were truly immunocompromised. 3% were on immunosuppressive medications, and less than 1% had an immunosuppressive disease. Sure, they had a majority of participants with significant medical problems and were therefore more at risk of bad disease outcomes, but truly immunocompromised individuals were basically not studied. The validity of the findings should be seriously questioned based on this discrepancy alone. I am disappointed that the study is being used as evidence to support the efficacy of a medication in immunocompromised hosts, and I find any interpretation of such to be fundamentally misleading. The second major fundamental flaw with this study can be deduced from the start and end dates during which the data was collected. The data was collected from November 2020 to May 2021. We know that Omicron did not become a variant of concern until December 2021, and it has quickly emerged as a predominant variant around the world and now makes up the vast majority of new cases. We also know that the Omicron variant has a relatively large number of mutations in the spike protein, and that the vaccines developed thus far are less effective towards Omicron when compared to other variants such as Alpha and Delta. The monoclonal antibodies, as discussed earlier, target the spike protein, so any mutations could quite possibly affect the effectiveness of such drugs. Since this study did not include data for Omicron, and since Omicron now causes the vast majority of infections, the validity of the study is again brought to question at the most basic level. Put all previous analyses aside. Now to my final thoughts. I can only come to the conclusion that the purchase of Evusheld from AstraZeneca by Canada was a decision based on bad science. On the surface, this research may seem legitimate in that it is randomized, placebo-controlled, double-blinded, and has a large sample size. However, on deeper analysis, the flaws become very apparent. First of all, the study doesn't even examine the population that it purports to represent, that being immunocompromised people. It also collects data before Omicron existed, and now that this is the predominant variant, it fails to account for any mutations in spike protein on the effectiveness of the monoclonal antibodies. Another huge note is that this paper is rife with conflicts of interests, which we disclosed earlier in the video. Putting these fundamental flaws aside, even if the results are extrapolated and applied to a different real-world scenario, the number needed to treat was low, and the clinical significance is highly questionable. Although the study purports to test safety, Phase 3 studies are not adequate enough to assess safety, and so we don't know the true safety profile. Based on this analysis, and on the fact that we now have effective medications for active disease that are known to be safe, such as fluvoxamine, for example, studies investigating the effect of these in immunocompromised hosts would be much more useful. Of course, studying generic medications would remove any financial opportunity for big pharma companies. So instead, US taxpayer dollars are helping fund the collaboration of the entire industry to develop and study new products that use relatively new pharmaceutical technologies and that have some kind of financial opportunities. In this case, the Canadian taxpayers are purchasing the medication before approval based on industry-sponsored research that lacks the basic essentials for practical benefits. For sure, the purchase of 100,000 doses by Canada is not huge news and bears little real-world implications, but ultimately, this is an example of the recent emergence of pharmaceutical companies as a major source of research being used to shape pandemic response strategies. Headlines in the mainstream present the results by using better-sounding relative risks, while I have yet to see the media report on scientifically useful metrics, such as the number needed to treat. Government decisions seem more and more guided by these reports of relative risk, rather than by the sound expert analysis of the methods behind those results, or quite simply by using the analysis of that, which is much more relevant. That begs the question, why have we been conditioned to put so much trust into industry research during COVID-19, and why are useful metrics of real-world applicability noticeably absent when reporting to the public? Again, we don't try to answer these questions on this channel, but these questions do arise. I wanted to end this video off topic with a simple observation that I'd like to share with the viewers. As a descendant of Egypt and as someone who visits often, I can tell you that this is an autocratic and non-democratic society. Some of the ways you can tell in Cairo are that government buildings are barricaded, perimeters are set up around them, there's a big police force, and security checkpoints run in the dozens. In addition, financial sanctions to government opposition are often imposed without court rulings, and opponents to the government are often jailed without due process. In the past few weeks, secondary to the trucker crisis, or what they call the Freedom Convoy, I have seen some eerily similar impositions within our society. Ottawa now has parliamentary buildings barricaded, perimeters have been set up around the city, there are now dozens of checkpoints, and a strong security and police force. In addition, financial sanctions on Canadian citizens were imposed without court rulings, and some of the organizers of the Freedom Convoy have been jailed and are not being given due process. To me, this is a concerning observation, and all political affiliations aside, we should try to prevent this from becoming the norm in our country. That being said, thanks so much for sticking around and watching the video. Please like, share, subscribe, comment below, and I'll respond to every comment. Please send to other people who you think might be interested so that we can collectively get better at analyzing science that the media reports on. Okay, see you on the next one.